Hi everyone, I welcome you all to the another lecture. So in the series of lectures on enzymes for physiotherapy, today we will be learning about the nomenclature and classification of enzymes. So previously जब हम पुरानी बात करते हैं, so there were tribal nomenclature of enzymes, and according to that tribal nomenclature, what was there? We put A's after the substrate. So it was just the tribal nomenclature, and later on, what happened? The International Union of Biochemistry in 1961, it classified the enzymes into 6 classes. The enzymes were classified into 6 classes, and every enzyme was given the Enzyme Commission number, the EC number. So the Enzyme Commission number was assigned to every enzyme, and the first digit of that Enzyme Commission number, it was designated for the main class. The first digit, it was designated for the main class. Okay. And moreover, enzymes are also classified into 2 groups. So this is not the official classification, but yes, according to the synthesis of the enzymes are broadly divided into 2 groups. One is the intracellular enzyme. In the intracellular enzymes, also known as endoenzymes, what is there? That the enzyme is acting in the same cell where it is synthesized. Where it is synthesized. It is acting at the same location. Okay. And from where do enzymes come? Enzymes are synthesized by the cell, and the information for the synthesis of enzyme, it is regulated at the gene level. The principle of central dogma, though recently it has changed that. We'll discuss this in some other class, but yes, the information for the synthesis of enzymes is located on the genes. So the enzymes which act at the site of synthesis, which act as the site of synthesis, are known as endoenzymes or intracellular enzymes. Okay, so one classical example of endoenzyme is the glycolytic enzyme hexokinase. It is acting within the cell and it is synthesized within the cell. And apart from these intracellular enzymes, the extracellular enzymes or the exoenzymes are the enzymes which act away from the site of synthesis. From the site of synthesis. So they are acting away from a site where they are synthesized. For example, pancreatic amylase. This pancreatic amylase, this is being synthesized in the pancreas, but whenever the action comes, it acts in the intestines. Okay, it is acting in the intestine. Okay, now moving on to the classification. Okay, moving on to the classification of enzymes. So enzymes are classified into 6 classes. Enzymes are classified into 6 classes by IUB, but nowadays there is another class of enzyme which is known as translocases. So इसको भी add कर दिया गया है, ठीक है? So these enzymes are of 7 types. Enzyme classification is of 7 types, and here is a short code to remember that. This short code is ortholyl. This short code is ऑर्थलिल एंड टी ट्रांसलोकेसेस के लिए जो हमने लास्ट में अभी ऐड किए हैं. O is the for oxidoreductases. T is for transferases. H is for hydrolases. L is for Lyases, I is for Isomerases, L is for Ligases, and the last enzyme, it stands for Translocase. It stands for Translocase. So still अभी जो old author की books हैं उसमें ये आपको नहीं मिलेगा. So you have to note that the classification of 7 types की है. ठीक है. तो अब हम सबसे पहले चलेंगे ऑक्सीडोरिडक्टेजेस की तरफ. ठीक है. सो ऑक्सीडोरिडक्टेजेस में जब हम बात करते हैं, अब आप देखो जो एंजाइम कमीशन नंबर की हम बात कर रहे थे, दैट एवरी एंजाइम हैज बीन गिवन एन एंजाइम कमीशन नंबर. तो उसमें क्या है, एंजाइम कमीशन जो फर्स्ट डिजिट है उसको दे दिया है ईसी. ईसी एंजाइम कमीशन फर्स्ट फॉर ऑक्सीडोरिडक्टेजेस. अगर पहला डिजिट 2 है, ट्रांसफरेजेस 3 है, हाइड्रोलेजेस 4 है, लाइएजेस 5, 6 और 7, 7. ऑल राइट. सो ये जो मेन क्लास है, ठीक है, एंजाइम कमीशन नंबर 1, इट इज गिवन फॉर ऑक्सीडोरिडक्टेजेस. अगर किसी के एंजाइम कमीशन नंबर 2 या 3 डिजिट का होता है, आप पहले डिजिट से पहचान सकते हैं कि वो एंजाइम कौन सा है. सो नाउ let us move on to enzyme commission number 1, which was oxidoreductases. Oxidoreductases. Now see, this oxidoreductases, as the name is suggesting, it catalyzes the reaction by oxidation and reduction. Okay, it catalyzes the oxidation reduction reactions. It catalyze the oxidation reduction reactions. ठीक है, ऑक्सीडेशन रिडक्शन रिएक्शंस सच एज, एक हमारे पास रिएक्शन है A plus B, it is converting into again A plus B. Here one substance is reduced and another one is oxidized. All right. So ऑक्सीडेशन रिडक्शन रिएक्शन के साथ जो एंजाइम को कैटेलाइज करता है उसको हम कहते हैं ऑक्सीडोरिडक्टेजेस. सो द एंजाइम्स इन दिस कैटेगरी इंक्लूड, व्हाट आर द एंजाइम्स? द एंजाइम्स आर डिहाइड्रोजिनेसेस, reductases, oxidases and peroxidases. Oxidases and peroxidases. All right. So now one example, as हम कह रहे थे dehydrogenase है, one example of this is alcohol dehydrogenase. So alcohol dehydrogenase, okay, and you can say lactate dehydrogenase. We already know lactate is converted into pyruvate, and this reaction is reversible with the help of enzyme LDH. LDH is what? Lactate dehydrogenase. Here electron carrier is NAD positive, which is being converted into NADH plus H positive. So this is the class first, oxidoreductases. Which was the second class? Second class was, we were talking about ortholyl. Second class was the transferases. Second class was transferases. Okay. So the transferases, you can say enzyme commission number 2. Okay. Enzyme class transferases. These transferases are the enzyme which catalyzes the group transfer reactions. Group transfer reactions. And they are named according to the type of group that has been transferred. Let's say if the amino group is being transferred, the enzyme is aminotransferase. If the amide group is being transferred, the enzyme is amidotransferase. And if the acyl group is being transferred, the enzyme is acyl transferase. Okay, the enzyme is acyl transferase. So what is going to happen here? One group is being transferred to the another. Let us say this A plus, in this A plus B reaction, A has a group X, and now in this reaction the enzyme catalyzes the reaction in which the X group it is transferred to the B. So A is liberated as such and this X group has been transferred to B. Okay, so I've already considered the names amino transferase, SGPT, SGOT. So these are the transferase enzyme. So the example is alanine amino transferase, ALT. Okay, so what this enzyme is going to do, alanine plus alpha ketoglutarate, it is being converted into pyruvate and glutamate. So this is a typical transamination reaction, and this reaction is catalyzed by alanine transferase, and here the pyridoxal phosphate, PLP, the pyridoxal phosphate, it is used as a coenzyme. This is used as a coenzyme. Okay, so let's move on to the next. Okay, third class was the hydrolases. Enzyme commission number 3, है ना? फर्स्ट डिजिट की हम बात कर रहे हैं. वी आर टॉकिंग अबाउट the enzyme commission number, the first digit of the enzyme commission number. So enzyme commission number 3, class 3, is the hydrolase. It is the hydrolase. So we already know that hydrolysis reaction is a reaction in which the bonds are cleaved by addition of water. So you can directly say it catalyzes, in simple language, it catalyzes the reaction by hydrolysis, All right, for example, AB plus water. Now hydrolysis of the bond AB, this is AOH plus BH. All right, examples are glucose 6 phosphatase. The enzyme is glucose 6 phosphatase. Okay, so what does glucose 6 phosphatase do? This glucose 6 phosphatase, it catalyzes the reaction, the conversion of glucose 6 phosphate to glucose in gluconeogenesis, अगर आपको याद हो, है ना? In gluconeogenesis, ठीक है? Okay, so this is glucose 6 phosphatase. Glucose plus 1 inorganic phosphate. Okay, so now ऑथलिल आपने याद रखना है. ऑथलिल याद रखने के बाद हम सीधे इस पे चलेंगे, ठीक है? Okay, so next is your enzyme commission number 4, which was the lyase. The enzyme commission number 4, see, let us take a quick recap. Enzyme commission number 1, oxidoreductases. Enzyme commission number 2, transferases, group transfer reaction. Enzyme commission number 3, hydrolases. It catalyzes the reaction by addition of water. Now comes the enzyme commission number 4. So this enzyme commission number 4 is lyase. This is lyase. This is lyase. Now this lyase enzyme this is going to lyse the bond, and when the bond will be lysed, it will be broken without water. Then hydrogen is not there, because the tetravalency of carbon, to fulfill the tetravalency of carbon, water is providing hydrogen. But when the lyase enzyme is being operated, the lyase enzyme works, it catalyzes the reaction by accompanying of double bond. So when the product is formed, the product will contain double bond. Let us give the example. Let us say this is AB and it has some groups X and Y. This is the substrate. This is being converted into product. So this A and B will contain double bond and this XY will be eliminated. Now this is the cleavage of bond without addition of water. So water contains hydrogen, तब डबल बॉन्ड नहीं आता अगर वाटर हाइड्रोजन देता, क्योंकि कार्बन की 4 वैलेंसी पूरी हो जाती. ठीक है. अब यहां पर क्या आएगा, यहां पर डबल बॉन्ड आएगा. तो इसका एक सबसे बढ़िया एग्जाम्पल हमारा है एल्डोलेज. So recall, where is the aldolase enzyme being used? It is being used in the glycolytic cycle. Fructose 1,6-bisphosphate it is being cleaved into two molecules. कौन से थे दो molecules? One was glyceraldehyde 3-phosphate. Another one was dihydroxyacetone phosphate. So aldolase is the classical example. Okay, aldolase is the classical example. Okay, now moving on to the next classification, which is the Enzyme Commission number 5, EC5. है ना, एंजाइम कमीशन नंबर 5 जिसको हम मेन क्लास कह रहे हैं ना. दिस इज आइसोमरेज. दिस इज आइसोमरेज. सो दिस इज गोइंग टू कैटलाइज द आइसोमराइजेशन रिएक्शंस. Isomerization reactions. All right. For example, ABC will be converted into CAB. So the classical example of this isomerase is glucose 6-phosphate isomerase. And this is going to catalyze the reaction conversion of glucose 6-phosphate into fructose 6-phosphate. Okay, so this is glucophosphoisomerase enzyme. Now the enzyme commission number 6. The enzyme commission number 6 is the ligase. Ligase enzymes are also known as synthetase. So this is important enzyme of recombinant DNA technology. When we introduce a gene of one plant into another and make transgene crop varieties, then the two DNA ends are joined by this enzyme ligase. फेविकॉल का काम करता है ना. What is this doing? This is joining, it joins the two ends of DNA, and with the help of ATP energy is required. Okay, जैसे ये मानो दो एंड है, 1 plus B, ATP is required. Okay, then AB plus ADP plus 1 inorganic phosphate. Okay, so next is last enzyme, translocase. So this enzyme helps in translocating the proteins, translocating the lipids, and translocating the ions across the membrane. across the membrane. All right. And the classical example of this is ATP synthase. The classical example of this is ATP synthase. This translocase is Enzyme Commission number main class 7. So that's all for today. We have completed the classification of enzymes. And in case of any queries, please let me know if there are any queries, and stay tuned for another lecture. Thank you.